Welcome to The Explainer. Today, we're diving into the incredible story that's written inside every single one of us, inside every living thing. From the way our cells divide to the reason some cats have that amazing patchwork coat, it all comes down to the fascinating world of genetics. All right, here's the plan. We're going to start with the fundamental rules of what I like to call the Great Genetic Shuffle. Then we'll see what happens when that shuffle goes wrong. After that, we'll look at a really curious case that seems to bend the rules entirely. And finally, we'll zoom way out to see how all of this plays out in entire populations. First up, the Great Genetic Shuffle. This is where we get into meiosis, which is it's like an elegant biological dance that ensures no two of us, aside from identical twins, of course, are ever exactly the same. It's the engine of variation. So, our bodies basically have two ways to divide cells. You've got mitosis, think of it like a photocopier. Its job is to make identical copies of cells for growth and repair. Same DNA every time. But then you have meiosis. This is all about reproduction. It's a reduction division, meaning it starts with a full set of DNA and ends up creating 4 totally unique cells, sperm or eggs, each with just half the genetic info. This whole thing happens in 2 big acts. Act 1, meiosis 1, is all about partnership and separation. The chromosomes you got from your mom pair up with the matching chromosomes from your dad. Then the cell divides, pulling those pairs apart, so each new cell gets 1 chromosome from each pair. But, and here's the cool part, which one it gets is totally random. This is the first big shuffle of the genetic deck. And then comes meiosis 2. This part's a lot more like that simple copy-paste of mitosis. The 2 cells we just made, they divide again. But this time, they're separating the identical copies of each chromosome, which we call sister chromatids. The grand finale? 4 genetically unique haploid gametes, each with its 23 chromosomes, ready for the possibility of creating a new life. So that's how it's supposed to go. But, biology isn't always perfect. It's a complex process. So what happens when that shuffle goes wrong? And this is a really big deal. This whole process is unbelievably complex. So sometimes things get stuck. A pair of chromosomes just doesn't separate like it should. And that one tiny mistake can have huge consequences down the line. Yeah, there's a word for this failure, non-disjunction. It literally just means not coming apart. And it can happen in either of those 2 big stages we talked about, either when the homologous pairs are supposed to separate or later when the sister chromatids are supposed to split. And that one little error kicks off this huge domino effect. The gametes at the end of the line end up with the wrong number of chromosomes. One might get an extra copy, while another is left missing one entirely. And this condition, the end result of that whole mess, having the wrong number of chromosomes, is called aneuploidy. It's super important to remember. Non-disjunction is the mistake. Aneuploidy is the result. It's the root cause of many genetic disorders. Probably the most well-known example of an aneuploidy is trisomy 21, which you might know as Down syndrome. This is what happens when one of those gametes with an extra copy of chromosome 21 gets fertilized. The resulting person then has 3 copies of that chromosome in every cell, instead of the usual 2. Okay, so we've seen the rules, and we've seen what happens when they break. Now let's look at something really cool that seems to bend the rules entirely. And for that, we need to talk about cats. You've probably heard this one, right? That calico and tortoiseshell cats are almost always female. Well, it's not just a fun fact. There's some seriously cool science behind it, and it's all hidden in their X chromosomes. It all comes down to this thing called dosage compensation. Just think about it for a sec. Females have 2 X chromosomes, XX. Males have an X and a Y. So to keep things fair and stop females from getting a double dose of all those X chromosome genes, nature came up with a pretty brilliant solution. Just switch one of them off. This whole idea is called the lion hypothesis. What happens is, really early in a female embryo's life, in every single cell, one of those 2 X chromosomes just gets randomly and permanently shut down. It squishes down into this tiny little thing called a Barr body. So what does that mean? It means adult females are literally genetic mosaics, a patchwork of cells, where some are using the X from mom and others are using the X from dad. And this is why we get calico cats. In cats, one of the genes for fur color is on the X chromosome. One version gives you orange fur, another gives you black. So if a female cat inherits 1 X for orange and 1 X for black, she becomes this living, breathing mosaic. In some patches of skin, the black X is turned off, so you get orange fur. In other patches, the orange X is off, and you get black fur. Amazing, right? Okay, so far we've been looking at genetics on the individual level, a single person, a single cat. But now let's zoom way out to an entire population. Because it turns out we can actually use math to describe and predict how all these genetic rules play out on a huge scale. And this is where the Hardy-Weinberg principle comes in. It's this core idea in population genetics that basically gives us a baseline. It says a population's genes will stay stable generation after generation, but only if a specific set of conditions are met. Big population, random mating, no mutation, no one moving in or out, and no natural selection. Now, of course, in the real world, these conditions are almost never met, and that difference, that's evolution in action. The principle gives us two really useful equations. The first one, P plus Q equals 1, is super simple. It just says the frequency of the dominant gene plus the frequency of the recessive gene has to add up to 100%. But the second one, P squared plus 2PQ plus Q squared equals 1, that's where the real power is. This lets us connect the frequency of genes to the frequency of actual genotypes. And that middle part, 2PQ, that's the one we're often interested in, the frequency of carriers. And this is not just some abstract math problem for a textbook. This has huge real-world applications, especially in something like genetic counseling, where it helps people understand their risks for having children with genetic disorders like Tay-Sachs disease. So let's take Tay-Sachs. If we know how common the disease is in a certain group of people, that's our Q-squared value, we can plug that into the Hardy-Weinberg equation and work backwards to find out how many people are carriers. That's the 2PQ value. And for some populations, the math tells us that about 1 in every 30 people is a carrier for the Tay-Sachs allele. That is incredibly important information if you're planning a family. So, look how far we've come, from the tiny shuffle of chromosomes inside a single cell, all the way to the mathematical odds playing out across millions of people. Genetics gives us this powerful lens to see where we came from and what might lie ahead. And it leaves us with one final big question. Now that we have the tools to calculate the odds of our own genetic lottery, what does that really mean for our future?